the previous video, we talked about an overview of metabolism. If you skipped that video, please make sure to check the link in the description below. In this video, we'll talk about the transport and fate of carbohydrates, proteins, and fats. So without any further ado, let's get started. Now, metabolic pathways can be studied under two different levels of organization. First one being the tissue and organ level, and the second one being the subcellular level. In the tissue and organ level, there is mainly transport of substances in between the different tissues and organs, whereas in subcellular level, there is transport in between the cytosol and the cell organelles, mainly mitochondria. Now we will focus on the subcellular level when we will individually deal with the metabolism of glucose, fatty acids, and so on. Here we will only study the tissue and organ level. Now let us start with glucose and amino acids. We'll quickly bring into the picture an intestine, a pair of kidneys, a skeletal muscle, and our liver, and the area between these being occupied by blood. Now, as you all know, dietary carbohydrates and proteins that we intake are hydrolyzed by the intestinal enzymes into glucose and amino acids, which is then taken up by the liver via the hepatic portal system. Zooming into the liver, the amino acids and glucose that enter the liver undergo the following fates. Now the amino acids can be converted to urea by the urea cycle and thus is eliminated by the kidneys to get rid of the harmful nitrogenous wastes. Or the amino acids may also be used for the synthesis of proteins via translation. The proteins then reach the systemic circulation, forming a part of the plasma proteins. Now during starvation, when the fatty acids and glucose reserves are used up, the amino acids can be broken down into glucose by gluconeogenesis, and then the glucose is released into the blood to maintain the plasma glucose level. The amino acids also are released in the systemic circulation. Now the glucose is mainly stored in the form of glycogen, which is synthesized from glucose by glycogenesis. Similarly, when the body needs glucose, the glycogen is broken down into glucose and the glucose released into the blood. Now inside the liver, the amino acids can be decarboxylated or the glucose can be oxidized to produce carbon dioxide. Okay, so now it may look a bit complex, but I hope you understand the major routes of the amino acids and glucose inside the liver. Let us shift our focus to the muscle. In the muscle, it takes up glucose from the blood and converts into glucose 6-phosphate, which has basically three fates. It can either be converted to glycogen, or it may be aerobically oxidized to carbon dioxide, or it may be anaerobically oxidized forming lactate. Now, in the blood there is the red blood cells. Now, since the RBCs lack mitochondria, they respire anaerobically, using the glucose in the blood, releasing lactate. Now, this lactate from the muscle and the RBCs reach the liver, and there they are converted to glucose by the enzyme lactate dehydrogenase. Now the pathway in which the lactate formed in the muscle goes to the liver to form glucose and is again cycled back into the muscle to form lactate is known as the Cori cycle. Now in cases of starvation, the glucose 6-phosphate can be converted to pyruvate, which is then transaminated to alanine using the enzyme alanine aminotransferase, which then moves into the liver to be converted to glucose via pyruvate. This is an adaptation during extreme starvation when the muscle is wasted for the formation of glucose for energy. Now this cycle of alanine, glucose and pyruvate in the muscle and the liver forms what is known as the glucose-alanine cycle or the Cahill cycle. The kidneys basically act to excrete the urea and the other harmful nitrogenous wastes from the body. Now let's talk about fatty acids. We'll quickly draw the intestine, skeletal muscle, the liver, and a specialized tissue, the adipose tissue, that stores fat. Dietary triglycerides that we intake are converted to monoglycerides and fatty acids for their absorption, which are then again converted to triacylglycerols after being absorbed. And then they are re-esterified with apolipoproteins to form chylomicrons, which are a type of lipoproteins. Now, what exactly are chylomicrons? What are they made of? We will discuss them separately when we will deal with fatty acid metabolism. Now, the chylomicrons can be taken up by the muscle after the triacylglycerols in it being hydrolyzed by the lipoprotein lipase or LPL into fatty acids, which can be either oxidized into CO2 or may be re-esterified into triglycerides, which is mainly the storage form of fats in the muscles. Now, this chylomicrons is only used by the peripheral tissues. For example, when it moves in the adipose tissue, it is again acted by the LPL or the lipoprotein lipase into fatty acids, which is again converted into triglycerides, and it is the storage form of fatty acids in the adipose tissue as well. Triglycerides or the endogenous triglycerides can also be formed from glucose via acetyl coenzyme A via lipogenesis. Now, during starvation, the fatty acids are released into blood in the form of free fatty acid or NEFA, that is non-esterified fatty acids. When the chylomicrons when the triglyceride contents of the chylomicrons is used up by the peripheral tissues, the remaining chylomicron is called the chylomicron remnant, which is taken up by the liver via receptor-mediated endocytosis, releasing its triglyceride content. Hence, the chylomicron basically serves to transport the dietary triglycerides from the intestine to the peripheral tissues. Now, the liver also synthesizes endogenous triglycerides from glucose and fatty acids that are derived from blood via the non-esterified fatty acids. Now, cholesterol synthesized in the liver, along with the triglyceride, combines with apolipoproteins to form VLDL or very low density lipoprotein. The VLDL serves to transport endogenous tag to the peripheral tissues, and in the process is converted to IDL and finally LDL, intermediate density lipoprotein and low density lipoprotein. These conversions form the so-called lipoprotein cascade pathway. Now, the LDL basically acts to transport the cholesterol from liver to peripheral tissues, and the cholesterol can be deposited in your arteries forming plaque. Therefore, it's atherogenic, which is why LDL is also known as bad cholesterol. The LDL and IDL finally reach the liver, and the cholesterol content in it is either recycled or excreted. Now, the cholesterol from peripheral tissues is brought back by another lipoprotein called the high density lipoprotein or HDL, which is why HDL is also known as a good cholesterol since it's non atherogenic. This is all. In the next video, we'll talk about glycolysis in detail. Hope you enjoyed it.